Meiosis is a type of cell division in sexually reproducing organisms that produces gametes, or sex cells. It involves two consecutive divisions of the nucleus, which eventually result in four haploid cells. The first division is called meiosis I, and it decreases in chromosome number from diploid to haploid, separating the homologous pairs of chromosomes. The second and final division is called meiosis II, where the sister chromatids are separated, resulting in four haploid gametes. Let's start with mitosis I. Interphase. A nuclear envelope encloses the nucleus. The nucleus has one or more nuclei and a single centrosome that is duplicated to form two centrosomes. Centrosomes are regions in animal cells that organize the microtubules in the mitotic spindle. Each centrosome has two centrioles. During interphase, the chromosomes duplicate. However, they are not yet visible because they have not yet condensed. In addition, the nucleolus becomes less and less visible during this phase. The first step is prophase I. Prophase is the longest phase of mitosis. Centrosome movement, spindle formation, and nuclear envelope breakdown occur as in mitosis. Chromosomes condense progressively throughout prophase I. During early prophase I, before the stage shown above, each chromosome pairs with its homologue, aligned gene by gene, and crossing over occurs. The DNA molecules of non-sister chromatids are broken by proteins and then rejoined. Each homologous pair has one or more X-shaped regions called chiasmata, where the crossovers have occurred. During this time, the nucleoli also disperse. Later in prophase I, after the stage shown above, spindle microtubules from one pole or another will attach to the two kinetochores, one at the centromere of each homologue. The two kinetochores, not visible yet, act as a single kinetochore. The homologous pairs then move towards the metaphase plate. Metaphase I. Homologous pairs line up at the metaphase plate with their kinetochores attached to spindle fibers from opposite poles. Anaphase I. The breakdown of proteins that are responsible for sister chromatid cohesion along chromatid arms allows homologues to separate. Each pair of homologous chromosomes, guided by the spindle apparatus, separates in anaphase I, with one homologue moving towards each pole. In anaphase I, the sister chromatids remain attached to the centromeres and move together towards opposite poles. A key difference between mitosis and meiosis is that sister chromatids remain joined after metaphase in meiosis I, whereas in mitosis they separate. The principle of independent assortment, Mendel's second law of genes for different traits assort independently of one another in the formation of gametes. Telophase I. In telophase I, a haploid set of chromosomes, each composed of two sister chromatids, usually containing some regions of non-sister chromatid DNA, reaches a pole. A nuclear envelope reforms around each chromosome set, and the spindle disappears and cytokinesis follows. Cytokinesis usually occurs during telophase I, forming two haploid daughter cells. In animal cells like these, a cleavage furrow forms. In plant cells, a cell plate forms. There is no replication of genetic material prior to the second division of meiosis. Meiosis II. Meiosis II is a shorter and simpler process than mitosis and can be thought of as mitosis for haploid cells. The cells that enter meiosis II are the products of meiosis I. In meiosis II, the sister chromatids separate, making haploid cells with non-duplicated chromosomes. Prophase II. The nuclear envelope breaks down, and the spindle fibers form to attach to the centromeres. Toward the end of prophase II, the chromosomes with both chromatids of the centromeres begin to move towards the metaphase plate. Metaphase II. The chromosomes align on the metaphase plate. However, each chromosome's two sister chromatids are not genetically identical as a result of the crossing over in meiosis I. The kinetochores attach to the microtubules from opposite poles of the cell. Anaphase II. In anaphase II, the proteins keeping the sister chromatids attached break down, allowing the chromatids to separate from their centromeres. Then, they move towards opposite poles of the cell as individual chromosomes. This is because of the principle of segregation, Mendel's first law, which dictates that two members of a gene pair, alleles, segregate from each other in the formations of gametes. Half the gametes carry one allele, and the other half carry the other allele. In telophase II, nuclei form, and the chromosomes begin to loosen and decondense. Cytokinesis then divides the cytoplasm in two, resulting in four haploid cells, each with an unduplicated set of chromosomes. These daughter cells are all genetically unique and not identical to their parent cells or each other. In spermatogenesis, 4 sperm are the result. Meiosis is complete.